Everybody all psyched up for that? Woo. Thank you. So we don't have a lot of material to cover to finish up translation, which is where the exam will cover through. So I'm going to finish that. And then rather than schedule a review session later tonight, which doesn't make much sense, I thought we would just do a review session after I finish this. So you can ask questions, and hopefully I can answer questions for you and get you going. I will say a little bit about the exam. I have written the exam, and the format is exactly that of the practice exam. I will strongly encourage you to look at that practice exam to make sure you understand the format, okay? So the first section, for example, is not multiple choice. Make sure you understand how that first section works. It's basically true-false. Circle if it's true, don't circle if it's false for each of the statements in the question. So it's important to take a look at that. If you think it's a multiple choice and you're going to pick one, that's not the way to go, right? So make sure you understand the format of that. The exam has 100 points. The first section has 16 points. The second section has 60 points, and the third section has 24 points. The third section is a longer set of answers that are more involved questions, and the second section is a short answer. So it's a pretty straightforward exam. Time should not be a factor. I do require you to finish in 50 minutes, okay? So, in fact, I'm kind of picky about this, and the reason I'm picky about it is I don't want some people having an advantage by taking more time. So my rule is when I say stop, you have to stop, because if you don't stop and I see you continuing to write, then you will lose points, okay? And that's just to be fair to everybody. I don't do it to be mean, but it's important that nobody have longer than 50 minutes, all right? I will also ask you when you come in to seat yourselves in every other location. So start at the end with number 1, all right? And then 3, 5, 7, 9, etc. Okay? If somebody's sitting in 2, don't sit in 4, because then everybody will have to get up. So sit in the right numbered seat. Okay? If you don't like a left-handed seat, then sit somewhere else. Okay? If you don't like a right-handed seat, then sit in a left-handed seat. Pretty straightforward. And as a left-hander, I spent my whole life writing on right-handed seats, so I know it's possible to have a seat that doesn't match your hand, if that happens, which I don't think it'll happen. And that's pretty much it. Any questions about format of the exam? Yeah. Will there be figures on the exam? It's a good question. There are no figures on the exam. Yeah. That isn't to say you might not use a figure to answer a question, but I have put no figures on the exam. Okay. Other questions? Yes. Yeah, good question. What's going to happen with the 2 quizzes that you've already taken? I think what I decided to do on those was to give you the higher of the 2. And that's I think what I'll do with that. So that's, I think, the fairest to everybody. Okay. Other questions? Yeah. Yeah, so can you draw an illustration to explain something? You certainly can. This is an effort in communication. So, if you use figures to explain something that you're doing, absolutely, that's certainly fine to do. What I would say is, however you answer a question, always make sure that you are communicating it to the best that you can. So if a figure helps you to do that, I think that's useful to do. I would also just encourage you to be precise. Okay? So commonly, students will come in and say, oh, I didn't get credit on this question, and I'll talk to them, and I'll discover that, well, they hadn't written something down, and they say, well, that's what they meant. And I'm sure you get tired of professors telling you, well, I can't give you credit for what you meant. I can give you credit for what you wrote. So be precise in what you write. I think that's an important thing to learn. One of the reasons that I emphasize lingo and language and so forth is that every discipline has its own lingo. So part of the base knowledge that you're acquiring in taking a class in a subject like biochemistry is you're learning some of that lingo, okay? And there's other things besides lingo that you learn. But I think if you're going to walk the walk, you need to talk the talk. And so understanding what those words and so forth mean is important. I've given you definitions. What is an operon? What is a this or that? So things like that, I think are important, because if you don't have a firm understanding of what those are, then other things you really can't make sense of. And so it's important to understand the words. And I know from last time that last lecture is one of the most complicated lectures I give. You heard a lot of terminology, and you heard a lot of things that sounded, similar. Elongation and EF and EFTU and IF2 and things like that. So be careful in going through these, and make sure, again, that you're understanding the differences between the different terms in the terminology. The Lac Operator and Operon system had an awful lot of things that sounded similar. So just be careful with that. And if you have questions, come see me. I should be around my office tomorrow. If you have questions tomorrow, I'm always happy to answer questions for you. Okay? Anything else? Okay. Well, let's finish up the translation for eukaryotic cells, and then we'll turn our attention to the review section. So last time I pretty much finished here talking about prokaryotes. There's really not anything to say about polysomes other than the fact that they're just simply places where multiple ribosomes are translating along a messenger RNA. So that's not any big deal there. And I've already talked somewhat about the eukaryotic messenger RNAs. So I'm going to remind you of the structure of eukaryotic messenger RNAs. All right. So eukaryotic messenger RNAs have two features that we don't see in prokaryotic messenger RNAs. They are at each end of the eukaryotic messenger RNA. So at the 5' end of the messenger RNA, we have something called a cap sequence. And the cap sequence is this unusual guanine nucleotide that is linked in a 5' to 5' orientation. It's the only place we find that in biology. And that prevents an exonuclease from chewing it up. So it's protecting the 5' end. And that turns out to be useful. And one of the things we see when we look at eukaryotic messenger RNAs is that they are around in a cell a lot longer than prokaryotic messenger RNAs. Okay, so the average half-life of a eukaryotic messenger RNA is in the order of about 24 hours. All right? In a prokaryote, that half-life might be 10 to 20 minutes. Okay? Well, this is also partly due to the fact that prokaryotic cells are dividing more rapidly than eukaryotic cells are. All right? So that's one difference. But the other is that because we have a longer time of cell cycle and cell division and so on and so forth, the needs for those proteins will occur over a longer period of time as well. So having some kind of stability in those messenger RNAs is useful. They're not completely stable because we still do want to break them down, and that does happen. The other difference we see in sequence of the eukaryotic messenger RNAs is at the 3' end, and the 3' end is actually fairly simply modified. It's called what we call polyadenylation, where a string of anywhere from 50 up to 200 or 300 A's excuse me, are linked covalently to the end. And like I said earlier, this also provides as a sort of a barrier against exonucleolytic degradation. An exonuclease that comes down and starts chewing up the end will go a little ways and fall off and go a little ways and fall off. So the more A's there are there, the longer it takes before it starts getting into the meat of the messenger RNA where the coding for the protein actually is. Now, as I noted in class when I talked about this previously, the cap and the polyA both play roles in the process of translation. And so I want to just show you a quick figure relevant to that. And that is right here. Okay. And what you see here is, well, it looks like a lot of things. I'm not going to throw all the things at you. But what you see is in green, you see a messenger RNA, a eukaryotic messenger RNA. So at one end, you have the cap. That's this 7mg that's down here. And at the 3' end, you have the polyA tail. Right? So they've kind of looped this guy around here. And that loop is appropriate because it turns out that during translation of messenger RNAs, the messenger RNA is held in a circular kind of structure like you see here using all of these various, and you see IF, 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 IF. These are initiation factors. So like we saw initiation factors in prokaryotic cells, we also have them in eukaryotic cells. In this case, they're helping to hold that messenger RNA into a circle. And that will play a role, as I'll say in just a second. You don't need to know the names of these. And the E stands for eukaryotic. Okay, again, you don't need to know that. I'm just telling you so you know. But you can see that there's a complex of proteins, one of which is binding to the polyA, and one of which is binding to the cap. So ultimately, the cap is, and the polyA are making this messenger RNA be present in a circular form. These caps and these initiation factors and so forth help to orient and bind the ribosome. Okay, they help to orient and bind the ribosome. So the ribosome sees this whole complex, excuse me, and goes on to it. Now, in contrast to what we saw in prokaryotic cells, eukaryotic cells have other differences in their messenger RNAs. Okay? One difference that they have is in bacterial cells, we have what we call operons, that is, multiple genes on one messenger RNA. Okay? The operon is, of course, a collection of genes under the control of a single promoter. So that means that one messenger RNA will contain several of these genes. That's in prokaryotic cells. In eukaryotic cells, there's one messenger RNA, one gene. So we don't have multiple genes on there. And that's important because remember that if we have multiple proteins or multiple protein coding sequences on a messenger RNA, we have to start it in the middle, right? So the ribosome has to know in prokaryotes where it is that the first AUG is, which is why we needed the Shine-Doe-Garneau sequence. Okay? So if there's something internal and there's a Shine-Doe-Garneau sequence there, the ribosome says, oh, there must be a gene here, and binds and then starts the translation process. That's not necessary in eukaryotic cells. Eukaryotic cells have one gene, and it turns out that eukaryotic cells have the first AUG is usually the one that's used to translate. So the ribosome can actually get on the end and start sliding until it sees the first AUG and then just starts translating from that point. That's not practical in prokaryotes. Okay? So the Shine-Doe-Garneau sequence is not present in eukaryotic messenger RNAs, and it's not needed. Okay? As I said before, the same genetic code is used to translate both prokaryotic and eukaryotic genes. So it's the same sequence. We have an AUG start codon, just like we have in prokaryotic cells. But one of the differences, another difference that we have in eukaryotic translation is the first amino acid that's put in is methionine, not 4-methionine. Now, I told you last time that in the case of prokaryotic cells, the very first amino acid put in is a formulated version of methionine. And that is necessary because the amino terminus of methionine is reactive, and when the second amino acid gets put in, if that amino terminus isn't covered up by something, then it'll cross-react and cause a problem. So the bacterial cells put a 4-mel group on there, which stops that cross-reaction from happening and allows translation to proceed. You might say, well, why don't eukaryotic cells have that? They've also got a methionine that's got a reactive amino group on its terminus. Why doesn't that happen in eukaryotic cells? And the answer is because eukaryotic cells have a ribosome with a slightly different structure, and it actually covers up that sequence. So it's not necessary to bind a 4-mel group to it. So consequently, the reactivity of that alpha amino group on methionine is not a problem, and translation can occur on just a regular old methionine. Okay, so the last thing is, well, why circular? Why is it that this is in a circular fashion? And people aren't completely sure about this, but one thought is that when a ribosome goes all the way around here and translates, and this is a stop codon here, and more likely the stop codon would be like up over here. But by the time it goes all the way around and translates, okay, when it's done, it's not too far away from the start codon. And so the thinking is that having this in a circle may facilitate reinitiation and translating of the same sequence by the same ribosome. If you have this only as a linear end, then one end is over here, the other end is way over here, and they're not as physically close to each other as they are when this thing is in a circular form. That's one thought about why this circularization is important in eukaryotic cells, but it doesn't occur in prokaryotic cells. Okay, questions about that? Yes? So does it mean the mRNA is being translated twice? In fact, most messenger RNAs are being translated many times. That's true both in prokaryotic cells and in eukaryotic cells. The difference is in prokaryotic cells, the translation is happening while transcription is occurring. In eukaryotic cells, this is happening in the cytoplasm. But again, this can be reinitiated by another ribosome, and it can happen many times. Yeah. As long as that intact messenger RNA is there, okay, that translation will continue to go on. So you'll have more translation that will occur. If you think about it, if a nuclease comes along and starts chewing up the polyA sequence, as soon as the polyA sequence is gone, there's not going to be anything that can bind to it, and then it can't circularize. And that circularization, if it's important for initiation, is going, this messenger RNA would have a reduced initiation frequency if it couldn't circularize. And so, again, as the polyA tail starts to shrink, it's going to be harder and harder for translation to occur on that messenger RNA, which, again, is important for control. And so cells want to control how much of a given protein that they're making. Okay, so that's the most complicated thing. And again, don't worry about the individual names of things there. That's not the important point. All right. Well, elongation proceeds in eukaryotic cells similar to what happens in prokaryotic cells. There are different factors with different names, and there are more factors that play roles in eukaryotic initiation than there are in eukaryotic elongation than there are in prokaryotic cells, but the names don't really matter. Okay, so for our purposes, elongation basically proceeds in the same way, and termination basically proceeds in the same way. We have these release factors called RFs, and I'm not sure if I gave that name in the lecture on Friday, but the things that terminate translation are called release factors, and I think I used the term RF, but RF stands for a release factor, and it's the release factor that brings the water into the ribosome that allows the breaking of the bond between the polypeptide chain and the transfer RNA. And the same thing happens in eukaryotic cells that happens in prokaryotic cells. We have the same stop codons in eukaryotic cells that we have in prokaryotic cells. So really no significant differences, for our purposes anyway, in terms of elongation and of termination. Okay, well, the last things to say are fairly brief, and they're interesting. One is a term that you will hear when we talk about proteins. It's called post-translational modification. And since translation refers to the synthesis of protein, this is referring to modifications that happen to a protein after it's been made. Okay? So things that happen to a protein after it's been made. One of the things that happens, and this is actually not at all uncommon, is proteolytic cleavage, meaning that a protease, which is an enzyme that breaks down or cuts bonds inside of a protein, can actually cut bonds inside of a protein and affect its overall shape and structure. What you see on the screen is a depiction of what happens to the hormone insulin. Insulin is a polypeptide chain, and it is obviously important for managing the levels of glucose that we have in our body. When we eat, our blood glucose levels rise, and as you will hear from me later in the term, glucose is a poison. And because the body considers it a poison, it modifies the level of glucose it allows in the bloodstream, and that modification is made possible by insulin. So insulin helps to control that. Now, that's not what the slide here is showing you. What the slide is showing you is that insulin is made in an interesting way. So we'll talk later about how insulin does what it does, but for right now, I want you to know how insulin is actually made. And the way that it's made is it's made as a fairly long chain. It's called pre-pro-insulin, which I don't even care if you know the name of that. The important thing about insulin is that it starts out as one long chain, And as I hope Dr. McFadden talked to you, there are sulfhydryl side chains that are present within that original long polypeptide chains. Sulfhydryls, again, as he probably told you, are reactive with each other, and they will form disulfide bonds, meaning sulfur-sulfur bonds. And these are important covalent bonds that are strong. They're much stronger than hydrogen bonds, for example. And when they form, they're very difficult to break, okay? Which is good because you want to hold the pieces together. And you'll see some of that happening here. So here's the sulfhydryls and the figure on the left. The middle figure here, you can see that the disulfide bonds have formed right here. So we see them holding here, and we see these two up here that are bonded to each other. The next thing, things that happen are there's two places in which this pre-proinsulin is cleaved by a protease, meaning it's broken by a protease. One of those is right here. All right, so we see the end of this guy is actually clipped off, and there you can see the cut site right there at this point. And you see this is shorter than that was. Okay? So this is the first of the post-translational modifications. A part of the protein is cut off. Okay? Now, why make it in this way? Well, I can't tell you the why, but I can tell you that it's not an uncommon way that proteins are made. They're synthesized, and then they're modified as necessary for the cell. The second thing that happens is there's a second place where the chain is cut. You can see the second sites where that triangle is there and down here, second and third if you want to count both. All right? And the result of those cuts leaves you behind with something that looks like this. So what we have floating in our bloodstream as insulin looks like it's two separate polypeptide chains. It actually started out as one, and it ended up being two because of the cleavages that I just described to you. If we don't have disulfide bonds holding this guy together, the chains would come apart, insulin would have no function, and we would not be able to use it to do the things that insulin does. So this is, as I said, not an uncommon set of post-translational modifications that will happen to a protein. We have digestive enzymes, for example, in our pancreas that are synthesized in an inactive form. And that inactive form is important because we don't want those enzymes. What those enzymes do is they're important in digestion. They break down proteins. And if we let them be active as soon as they're made, then what will happen is they will start attacking the cells that made them. And if you start attacking, it's kind of like biting the hand that feeds you. If you start attacking the cells that made this, then you're going to have a real problem because the cells may die, et cetera, et cetera. And that is a factor in a disease called pancreatitis. So they're synthesized in an inactive form. I'm just telling you this, by the way. You don't need to know this. They're synthesized in an inactive form. And then later, a protease, as they get into your digestive system, a protease will cut a specific bond within them and activate them so that they're active in the place where you want them, which is in your digestive system, and not in the place where they were made. So that's just an example of why sometimes these post-translational modifications can be very helpful and important. Okay. Let's see. So that's that. Another thing that happens is the cells have a way of basically keeping house. Keeping house is important, okay? We saw that messenger RNAs, for example, had nucleases that were important for breaking down messenger RNAs. So we saw that there was a lifetime. They were synthesized, they were used, and they were broken down. Well, proteins are similar to that. They are synthesized, they are used, and then they too are broken down. In the case of proteins, they're frequently broken down because they've gotten damaged. Okay? They've gotten damaged during the process. Well, we don't want to have a lot of damaged proteins floating around in our cells. So cells have a sort of a recycling bin, if you want to think about it that way, called a proteasome. Proteasome. A proteasome is basically a little chamber that breaks down proteins. So like we saw exonucleases would break down messenger RNAs, so too will proteasomes break down proteins over time. And it's actually a rather complicated process that I'm not going to go in, I'm not going to go into or hold you responsible for. But suffice it to say, the proteasome plays a very important role in recycling proteins within a cell. Okay. The last two things I want to say about translation relate to a couple of interesting things. One is something we call the 21st amino acid. The 21st amino acid literally is that. It's actually used, it's not something that's made by an alteration of an amino acid after a protein is made, but instead is incorporated into a protein during the time it's being synthesized. It's an odd mechanism that the cell uses, but it's not unknown. You have, for example, this amino acid in some of the proteins in your body. Okay? Well, how does it get in there? We've already seen the genetic code, and the genetic code doesn't have anything for selenoscysteine. How does it make it into there? Well, it turns out that there's an odd transfer RNA that is made that can pair with one of the stop codons. An odd one. It's not common, all right? And that transfer RNA can be linked to a selenoscysteine. So under the right circumstances, this can actually insert selenoscysteine into a protein that's being made. And some of the proteins that we have in our body actually require having selenoscysteine put into them. So it's not a common thing, but it's also not an unknown thing. We have a few of these in our body that make this. And selenoscysteine is definitely something that would be the 21st amino acid, but it's not one of the standard ones called for by the genetic code. All right, and the last thing I will say something about with respect to proteins is on this figure, and this is a pretty important thing. This is a collection of proteins called chaperones. Okay, chaperones. When I was a kid, we were never, if you were under 16 and you wanted to go out with a girl, or a girl wanted to go out with a guy, or whatever, you had to have your parents chaperone you, right? So the parents drove you around and you went and did this, and it was embarrassing for all concerned, okay? And chaperones were there to kind of make sure everybody was, I guess, behaving themselves, as it were, all right? Well, among proteins, the same sort of phenomenon occurs because proteins, as Dr. McFadden probably told you, have to fold properly in order to function properly. If proteins don't fold properly, then there can be some pretty significant consequences that can arise. So, for example, there's a disease that I'm sure everybody's heard of called mad cow disease. And mad cow disease arises as a result of a protein in the brain that doesn't fold properly. And not only does it not fold properly, but because once it gets unfolded or misfolded, as that happens with this particular protein, it induces other copies of the same protein to also misfold. So that really, if you think about it, causes some pretty drastic effects pretty quickly, because one protein affecting the folding of an identical protein in the wrong way now multiplies and multiplies and multiplies pretty quickly. So mad cow disease is the disease that arises from that. Cattle that have mad cow disease usually succumb to it very quickly. There's a human disease that's similar to it. It's called Creutzfeldt-Jakob, and no, you don't need to know that. That has the same phenomenon. That is, there's a misfolded protein in our brains, and when that starts misfolding, people who get this die of the fact basically that their brain doesn't function, and they don't last very long either. The same sort of thing happens. There's this type of misfolding is prevalent throughout all of the animal kingdom. Sheep get a disease called scrapie, which is also very similar to this. Anyway, the importance I'm trying to tell you of this is that folding properly is important. And some proteins have difficulty folding properly. This may happen because of the structure of the protein. It may happen because maybe you got a fever. Okay? So getting a fever, for example, can raise your body temperature a little bit. And one of the things you should have seen about the structure of proteins is that temperature is a factor in protein unfolding. So making sure that they fold properly can be a factor, and that's where the chaperones come in. So there are a couple proteins. One's called GRO-L, the other's called GRO-S. These are in bacteria, but we have similar ones in our body. that allow a protein, basically a chamber in which to fold. A chamber in which to fold. Okay? Some of the misfolding that happens can happen because a protein, as it's being synthesized, may start interacting with another protein, and that interaction can cause a problem. The Creutzfeldt-Jakob or mad cow disease would be an example of that. Okay? If you can isolate the protein and allow it to fold on its own, it's more likely to fold properly, and that's where the chaperones come in. So chaperones are very important for not all proteins, but for some proteins to allow them to fold properly. Clear as mud? Well, believe it or not, that's the end of the material for this exam. So I have a song. Would you guys like a song now, or would you like to do the review first? Song? Okay. This one is about something, I was sitting in your seats, all right? It's many years ago, mind you, but I was sitting in your seats having exactly the same kinds of thoughts that you did. And one of the things that you have as a student is you have student nightmares, right? You wake up, and you're in the middle of the night, you're having this dream, and oh my God, I forgot to take this class, and it's halfway through the term. I still have student nightmares, all right? Sometimes I have professor nightmares where I forgot I'm teaching this class, and I don't know anything about the subject. Okay, that's one that I have. And the other is, oh my God, I'm teaching this class, and I forgot to go to class, and blah, 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 and I don't want to go into that. You guys probably know something about that. All right. So what I did was I had this idea, and so I wanted to encapsulate that in a song that I call Student Nightmares. So give me a second to get this set up, and I will show it to you. It's also one that I can't sing very well, so I've got somebody to record it. So a Beatles tune. Answered 3B, but then I thought it might be C. So I hope you guys don't have that nightmare tonight now that I put the idea in your heads. All right. So let me get back over here. And the way these work, my review sessions, they're driven by you. So you ask me questions, and I'm happy to try to answer them. Yes? Okay, so what would you like to know about the Lac Operon? So I'd like to have kind of specific questions. That's fine. Okay, so let's go over the Lac repressor. Okay, let's take a look at the Lac Operon, and let's see, be about right here, and the Lac Operon structure is right there. Okay. So this is the figure that I started talking about the Lac Operon. And the Lac Operon, so let's get some terms down here, right? So an operon, of course, is a collection of genes under the control of a single promoter. So this is showing the control region, which is the promoter region, for these genes. There are 3 genes in the Lac Operon, which means that when the messenger RNA is made, it's going to contain those 3 genes. Again, this is a prokaryotic cell, so prokaryotes can have more than 1 gene on a messenger RNA. All right, so the operon is, this is the 5' end. The operon extends way out here off to the right. The operator is basically the promoter region. Okay? If you think about it as the promoter, that would be fine. All right? The operator is basically the promoter region of the operon. All right? Well, promoter contains a -10 sequence, contains a -35 sequence. -10 is called a Pribnow box. And it's at that operator region where 2 different things can bind. One of those things is the Lac repressor. Okay? If the Lac repressor binds to the Lac operator, the RNA polymerase cannot bind. Period. Okay? If the Lac repressor binds, the RNA polymerase cannot bind. If the RNA polymerase can't bind, no transcription. Okay? The question then is, well, when does the Lac repressor bind to the operator? And the answer is whenever there's no lactose. When there's no lactose, the repressor, the Lac repressor, will bind to the Lac operator, and there will not be transcription. That makes sense, because we don't want to make those genes unless there's lactose. Not we, bacteria. I always say we. We as a living organism of which bacteria are there, right? Okay. If lactose is present, however, what will happen is the Lac repressor will bind to lactose, and when the Lac repressor binds to lactose, it can't bind the operator anymore. That means that now the operator is open for business. The RNA polymerase can bind, and the RNA polymerase can start transcription. Now, I noted in class that the promoter, that is the -10 sequence of the promoter, was not a very good -10 sequence. It wasn't a very strong promoter, which means that the cell wasn't going to make very much of that messenger RNA. However, when there's a lot of lactose around, then there will be synthesized cyclic AMP, which I call CAMP, C-A-M-P. Okay? And when CAMP is present, it will bind to a protein called CAP. So you have CAMP, which is a molecule, CAP, which is a protein, and CAP, when it's bound to CAMP, will bind to the CAP region of the Lac Operon. All right? When CAP binds here, it favors the binding of RNA polymerase over here and enhances transcription, meaning it makes it much more likely transcription is going to happen, and consequently much more of the operon will be made. Well, what's in that operon? What are the genes that are in that operon? 2 of them we don't really worry about for our purposes, but one is very important. It's called beta-galactosidase. And beta-galactosidase, we'll talk about it actually next week in another context. Beta-galactosidase breaks down lactose. Right? It breaks down lactose. And so that's very important because once the cell has lactose in it, it wants to use that lactose for energy. Does that help? Yeah. Okay. Did you have other questions with that? I'm sorry? No, it's not the same as GLUT. No. So GLUT is a glucose transport protein. So GLUT is used to move glucose into a cell. That's not a GLUT. No, different thing. Okay? Yes, question?
And where do you say the CAP comes from?
The CAP is a protein that's being made by the cell. The CAP is being made when there's more lactose present. Okay? So there are enzymes that will synthesize CAMP. We'll talk about those a little bit later. But CAMP is used, and it's used in our cells as well for different purposes, but CAMP is used for the purpose we call signaling, meaning that it's signaling something, some kind of a message or some kind of a piece of information to a cell. Yes, Jane? Is more CAMP made when there's more lactose? From a simple perspective, the answer is yes. Okay? Yes? So when CAMP binds to CAP, then CAP can bind to the CAP binding site. That's correct. And if there's no CAMP, then the CAP cannot bind to the CAP binding site. Yes?
So then, like, how come, like, when there's high level, like, wouldn't that mean that it's still expressed, like, the beta lactose, even if the, even if there's no glucose present? Like, doesn't it only express it?
Okay, so remember this, it's a beta-galactosidase is being made in response to lactose, not to glucose.
Yeah.
So I'm not sure I understand the question.
And I thought you needed low levels of glucose for it to be expressed as well. It does better as a source of energy.
So it's, well, your question is, it's actually, the answer to your question is a little complicated. So his question is, if glucose is present, how will this affect things, which is basically your question. Glucose will affect this, and that's why CAMP is actually important. So I'm giving you a simple picture of CAMP. CAMP is actually indicating the energy level of the cell. So if cells have very low levels of glucose, one of the things that will happen is they will start making cyclic AMP. So that does affect the picture. I haven't said that because I didn't want to get people confused about that. I'm trying to keep the information simple, but you are correct in that. Okay? Believe it or not, I do try to simplify things for you guys. After that lecture on Friday, you're kind of going, no way, man. Yes? Anything else left on Lac Repressor? Lac Operon? Everybody goes, oh, ask a question. What about? Okay, so basically the initiation and elongation phases of translation. Did you have specific things about that you wanted to know? Okay. Well, okay. Well, let's look at this. His question is, what's the function of these, et cetera. So let's try to wade through some of the nomenclature from the lecture on Friday. Okay. So from a big picture, I want you to stand back and think about the process of translation as occurring in three main steps: initiation, elongation, and termination. We had 3 steps to transcription. We have 3 steps to translation. And I said we could add a 0 step where we talk about the addition of the amino acid to the transfer RNA. Okay? Now, initiation, elongation, and termination. In each case, there are proteins that help these processes to occur. All right? What has to happen during the initiation phase is that the cell has to assemble the ribosome on the messenger RNA in the right place. That's what has to happen in initiation. It has to assemble the ribosome on the messenger RNA in the right place. All right? So the proteins that I've described to you help that to occur. All right? So we start out with the ribosome in 2 pieces. One piece called the small subunit, which has a bunch of proteins and a ribosomal RNA called the 16S in it. And we have a large subunit as well, and that's shown over here, called the 50S. So we have a 30S ribosomal subunit and a 50S ribosomal subunit. Each subunit has multiple proteins. Each of those contains at least one ribosomal RNA. The 30S ribosome contains something called the 16S ribosomal RNA. Excuse me. And that is important for the alignment purpose that I talked about with putting the ribosome in the right place. All right? To get the process started, there's something called initiation factor 2, IF2, the number 2. Okay? Initiation factor 2 carries to this messenger RNA and the small subunit, it carries the initiator tRNA, meaning it's got a 4' methionine attached to one end of this tRNA. IF2 is covering up the bond between the amino acid and the tRNA because, again, this is occurring in water, and water will break that bond. IF2, all right, facilitates the joining of the small subunit to the messenger RNA, and more specifically, it aligns the tRNA to the start codon, AUG. Well, how does the right start codon appear? How does the right AUG appear there? That happens as a result of a sequence in the messenger RNA forming base pairs with the 16S ribosomal RNA in the small subunit. Okay? So here's the Shine-Dograno sequence that is going to form base pairs with the ribosomal RNA. When it does that, the AUG is aligned in exactly the right place for translation to begin. Okay. There are two other initiation factors I didn't talk about in class, but since you've asked the question, I'll bring it up, and no, I won't hold you responsible for them. They're called IF1 and IF3, and they also help facilitate this process. All right, once we have put this beginning complex together, the only thing that's left is we need to add the large subunit to it, and that creates that snowman-like structure that I described with a big subunit and a small subunit. And in between the two, that's where we have the messenger RNA. So during the process of translation, this snowman structure is going to go sliding down the messenger RNA, reading the codons and putting the proper amino acid in as a result of that. That happens in elongation. At this point, we have just finished termination. At the end of termination, we have the snowman structure. We have the messenger RNAs in there. We have the start codon forming base pairs with the AUG. And we're ready to go to the next phase. Yeah. Okay, there's a lot. There's a lot there. Before I go to the next phase, I'll take any questions or clarifications that you'd like from me. Yes?
Is it the 50S and the 30S that contains the A site?
Very good question. So the 30S and the 50S together have a chamber, have three different chambers. The A site, the P site, and the E site. That's correct. At this point, the initiator tRNA is in the P site. And just for your information, again, you're not required to know this, but the initiator is the only one that first starts in the P site. All the other ones come in through the A site because that's where they come in during elongation. Okay, so we've now finished initiation. We turn our attention to elongation. Okay, so elongation occurs, and I wish these figures were bigger. I apologize that they're not. Initiation occurs. We can now see, here's the snowman structure. It's not drawn very much like a snowman, but it really is. A site, P site, E site. Messenger RNA going from 5' on the left to 3' on the right. Excuse me. And now we've got a codon sitting in the A site, and there's no tRNA there. So that is a prime place for a transfer RNA to come in. A transfer RNA comes in at that point, okay, and forms base pairs with the codon that's in the A site. That transfer RNA is brought in by the protein called EFTU. That's the other protein that's covering up the bond between the amino acid and the transfer RNA. Well, now we have the two transfer RNAs and the two amino acids sitting right next to each other. All that has to happen at this point is that the two amino acids have to be joined together by a peptide bond. And that's catalyzed by a ribozyme, which is a catalytically active RNA, which turns out to be the 23S ribosomal RNA that's in the large subunit. So it catalyzes the joining together of those amino acids. We now see that they're both linked to the tRNA that's in the A site, and we're ready for what's called translocation, which is where this protein called EFG uses the energy of GTP to slide this whole thing down by one codon. Yes? GTP is used almost, well, GTP is the only energy source used in the process of translation. There are a few other reactions where GTP is used in the cell. A few, but not many. Primarily used in translation. Okay, so that is elongation. Other questions on elongation? As you know, the termination is the simplest. Yes? Yes. It's moving towards the 3' end. That's correct. So everything's moving from 5' to 3'. Yep. Olivia.
Is it the ribosome that reads the codon in the A site and finds the anticodon?
Good question. So she asked, is it the ribosome that is reading the codon in the A site and specifying which tRNA is going to come in? The actual reading is actually done by EFTU. EFTU reads that and makes sure that the base pairs are proper. You don't need to know that, but that's just to answer your question. Yeah, okay? I keep saying you don't need to know that. You say, well, I don't have to know anything, right? So this is a good deal. Ahern said, I don't need to know anything, right? Was there another question? You got it? Okay. Okay, so let's turn our attention then to termination, and termination is actually the simplest of those. In termination, these proteins called release factors come in. Well, they come in only when a stop codon appears in the A site. Only when a stop codon appears. When a stop codon appears there, is there a class in here at 5? I sure hope not, but I see people standing up front. Oh, blast it. Oh, well, I'll try to get through questions quickly. Okay. The replication, the release factors come in, and they carry water. So when there's a stop codon in the A site, the release factor comes in, carries water, and water breaks the bond between that polypeptide chain and the tRNA that's in the P site. The polypeptide chain goes away, and everything falls apart, and the whole process is done. Clear as mud? There's a lot of names there. A lot of names there. Okay? I can assure you that I like to look at this in terms of the big picture. Okay? So I try to avoid too much detail. Okay? But I do also think that this is a process that's important. So you can use that judgment as best you see fit. Yes? Uh-huh. Okay, good question. So her question is, when the tRNA leaves the E site, what happens to it? Well, actually what happens is that tRNA has lost its amino acid, and so that enzyme I call Larry comes and attaches a new amino acid to that tRNA, and it's reused. Yes? You can call Larry on the test, yes. Yeah. About what? Okay. Say it again. I can't quite hear you. There are change what? Chain terminators. Okay, yes, I'm sorry, yes. Yes. Okay, so what she's asking about is the figure that I had, back to replication, which is here. And if I look at, where was it? Oh, actually, maybe it wasn't in replication. It was actually in the structure. So if I go back to here, tides, RNA chain, base pairing, flipping, supercoiling, denaturation. It's there somewhere. That's not it. anyway, to try to answer your question, chain terminators are things that I described that don't have a hydroxyl on position number 3. All right? So these are man-made compounds. These are not found in eukaryotic cells. They're not found in prokaryotic cells. We're introducing them as drugs into a cell. So we could use these on a bacterial cell if we wanted to. It would be uncommon to do that, but we could do it on a bacterial cell. Most commonly, we're doing it with respect to viruses that affect humans. That's the most common place that they're used. Okay? Yes? The function of the leucine zipper is actually, that's a good question. What leucine zipper is, if we look at a protein, and I showed you how all the leucines can align on one side, okay? On the same side, you'll see those leucines about every 7 amino acid residues. What happens is if I have another one over here and one over here, the leucines actually do form like a zipper like that. That's why they call it the zipper. And they help to hold 2 strands together. That's the function of a leucine zipper. That's how they actually work. I apologize for not knowing there was a class in here. I didn't think there was anything in here after we were. So, anything else? I will say this, since I am cutting this a little short, I'll give you my cell phone number. If you have a question tonight, give me a holler, okay? 541-908-4465. And I'll